This is the prep for lecture 13, and I'm going to try to cover all the sea urchin development in just one lecture. I want to make sure we have lots of time for the vertebrate work, which I'm pretty sure students will be more interested in. But sea urchins, there's a lot to discuss, so I'll do the best I can to hit just the most important topics. So sea urchins, we've already looked at a little bit because we covered sea urchin fertilization in lecture 5 as an example of organisms with external fertilization. And we're going to spend a little bit of time speaking just generally about the early embryogenesis. We'll talk about where the cells lose their totipotency. The first few divisions are equivalent, but then there's asymmetric divisions that begin after that. Asymmetric cell divisions is as you know, a repeating theme in the class. And then we'll be talking about the fate map, so the blastula that has cells that are already specified to form certain structures. They're not determined yet, but they are specified, and if nothing is disturbed, they will go ahead and form their particular structures. And we'll spend a lot of time talking about signaling in this part of the embryo. And then we'll talk about the axes as well. These organisms have what are called animal vegetal axes, and then oral, that means the mouth, and the aboral, or that means not the mouth. Very imaginative names. And so the axes, they're a little different. They're named differently. This is a radially symmetrical animal, so it's hard to imagine an anterior or a posterior. But the fact is that if you look at the animal and the vegetal, the genes involved correspond to the genes required for anterior and posterior specification in other organisms, so the same kinds of signaling molecules and so on. And we see that sort of here as well. And then the oral and aboral is similar to sort of ventral and dorsal signaling when we look at the actual some of the genes involved. I'm going to keep the genes to a minimum. There's a lot of information in this chapter. It's pretty cool, and I think that Dr. Hart studies this topic, so we have someone in the department who does work on this field. Okay. So this is the basic life cycle. So the adults are separate, so they're separate sexes, and the females and the males at a certain season there are triggers, and they release gametes, tons of them, into the water. And the fertilization process we covered just a little bit in lecture 5. After fertilization, you should know what this structure is around here. And there are a few fairly rapid cell divisions. A lot of embryogenesis is fairly quick because it's a vulnerable stage, especially the earliest stages. In these organisms, the gastrula and the others are swimming. They're able to move around, but these guys are pretty vulnerable. Someone could come along and eat them anytime. They're just kind of floating. They have no cilia, and so and so we'll talk about the first few divisions. We'll talk about the fate map in the blastula, and I will talk just a little bit about the movements that occur during gastrulation because they're kind of interesting. And then we won't really be talking about the rest. After gastrulation, when the ectoderm is on the outside, the endoderm, the gut, is in the tube that goes through the body, and then the mesoderm is basically skeletal elements that are found in between. After that, hatching into the larvae, they swim freely for quite a bit of time before they find their way to rocks in intertidal zones most of the time. and they set down on those and go through metamorphosis. Okay, so we're going to talk a little bit about this and the specification of the axes, the oral versus the aboral, and we'll talk about the signaling that takes place, as it turns out, from the vegetal cells up towards the animal cells on top, and how a little bit about the specification of the tissues. Okay, so this is up to the blastula stage, and as I said, the first two divisions are what we would call equivalent divisions. They're at right angles to each other, and they slice down from the animal to the vegetal. So all the cells now have whatever molecules are present, in the vegetal region, and whatever is in the animal, and whatever's in between. They have everything. After the third division, where we have the 8-cell stage, now we have cells on top that don't have the same information, the complete information, and cells below that also don't have complete information. So they're no longer totipotent, basically. And then after the third division, the cell divisions are no longer all at the same angle. So, we had 4 columnar cells, and they all had a cell division, with the same axis. That doesn't happen. The top 4 cells divide sort of this way to make a ring of 8 cells. And then the bottom cells, they divide in this weird, really asymmetrical way, where you've got, and it's still on the same plane as the previous one, where you have some very small cells called micromeres at the bottom, and then the cells that the other cell that results are bigger than usual. And so these are called macromeres because they're big ones, micromeres because they're small ones, and then these are referred to as mesomeres. Meso means in between or medium, we would use that. And then this continues to divide. Over time, when we get to the 64-cell blastula stage, the cells are not as hugely different. These guys are still a little bit bigger, but because of the way the subsequent divisions have taken place, they're pretty much the same size. The only thing that is still unusual is that you still have these really tiny micromeres down here. And you have this region which is called the vegetal plate, and it's composed of the 2 layers of cells that are directly above the micromeres. The micromeres are specified to form mesoderm. They're colored in red. The next layer of cells up here forms both endoderm and mesoderm, so it's referred to as endomesoderm. When they divide one more time, the cells on top that result will be specific for endoderm, and the ones below will be specific for mesoderm. But there's a really interesting process of specification there. And then these cells have endoderm specific in the bottom, and ectoderm specific in the top. And then again, after the division takes place, this row of cells is going to contribute to that endoderm, the gut. So that's this part. And then this will be the ciliated ectoderm, which is the sort of like the skin on the outside of the larva that has cilia on it, and it helps them move around here. And then up towards the very top is where the neuroectoderm, so the nerve tissues, will be. Okay. Yeah, and just take note of the sort of the fate map. It's kind of colored in so that you can see, which cells are going to give rise to which structures. Okay. And so the cells are at this 64-cell stage are very precisely specified. And this basically shows what I just finished talking about. After gastrulation, we have this is a late gastrula. And you can see that all the ectoderm is on the outside. The pale blue on this side is the aboral ectoderm, and this is the oral ectoderm because it's on the same side as the mouth. Here's the gut inside, and a few cells here that form this structure called the oral hood. So it's just part of the, like, where the food gets taken in. And then there are these are called primary mesenchyme cells. Mesenchyme cells are loose cells that the cells in this part of the body, they're sort of nicely arrayed, one right next to the other, and they're held together by little junctions that hold them together. And the cells, these ones, the secondary mesenchyme, and these ones, the primary mesenchyme, these are cells that have let go of their neighbors and rounded up, changed shape a little bit, and then migrated inside. And so, and they form these sort of kind of loose tissues, so they're not all stuck together. And these will form spicules, which are the skeletal structures of the little Pluteus larva. So, and as I said, the cells are specified at this stage. Many diagrams call it the 64 cell stage, but I believe there's actually only 60 cells, so it's also frequently shown as the 60 cell stage, but they are not yet determined. Okay, and so then I want to talk just a little bit about the gastrulation process because it's really interesting, but we won't do any more than just this one slide. So the cells on what's called the vegetal plate, they get kind of thick. They're a little bit thicker and sort of very tightly connected compared to the rest of the tissues. These are thinner. They're still connected to each other. They form what's called epithelium, which is cells that are all connected and they're tightly connected. But these guys kind of scrunch up even more. And then the primary mesenchyme cells, the mesoderm specific ones that came directly from the micromeres, those ones kind of migrate in. But they migrate in, and they, at the beginning of that migration, they sort of, they're kind of congregated, gathered atop the endoderm cells here. And something induces a change in shape of these cells so that, remember, if you narrow one end of the cells and they stay connected to each other, you can wind up making a sort of a groove. And that's exactly what happens here. This starts to invaginate inwards. And so that occurs, and then the other cells kind of move in and around, and you form this archenteron structure, which is the beginning of the gut. We'll hear about this structure in other embryos as well. And then also, the actual invagination and migration process only gets them partway there. Then the cells, they send out these long processes that reach and touch the roof of the gastrula on the inside, and they're seeking the right place, because this is the anus, and it's going to try to find the spot where it forms the mouth, so that you have the tube that goes right through the body, right? And so it's looking for signals on here. Now, we don't know exactly what the signal is, but I'll talk about oral, aboral specification towards the end. In any case, these little processes are sent out, but eventually they manage to connect to the right place, and then there's a kind of contraction. So the cells, they're kind of pulled upwards and then over to the side, and that's what you see. This is ends up with the gut pulling kind of upwards, but then when the process reaches the correct tissue, the correct signal, sorry, then it, this all bends over and forms the mouth, right? And so, that's why this is called the oral side, and that's why this is called the aboral side. And then, as I said, these paler yellow, these are secondary mesenchyme. They have migrated, pulled away from the rest of the gut and formed these separated cells, sort of looser kind of cells, but they will congregate eventually and form the oral hood, which is just a part of the structure where the food kind of comes in when the organism is eating. And there's all these cilia around here that help the food in to where it gets eaten up. And then these primary mesenchyme, mesodermal derivatives, they actually form these spicule structures. So they're skeletal rods, they're sometimes called, and they give strength and shape to the little larva. Okay, so that's all we're going to say about the gastrulation process, but it is pretty cool. So as I said, the first two divisions are equivalent, so it's harder to see when we see them sort of from these different angles. So I showed the schematics first, but the idea is that the first, if we look at, so this is the expression of a particular gene, and the first cell division, divides the embryo into two cells that are still both expressing this gene. And then the angle is kind of sort of peculiar. The embryo is a bit flattened, but these are now 4 cells, and they still all have one surface that's expressing that gene, which is on the vegetal side of the embryo. And they all have as well whatever genes are being expressed or whatever proteins are present on the other side. So those, the cells all have the same information still. So when we say cell divisions are equivalent, that's what we mean. The cells that result, they have exactly the same fate as the original cell. So if this had the ability to form the whole embryo, each of these does, and each of these does. And that's what's shown here. So this may remind you of lecture 2, where we talked about Hans Driesch's experiments with the technology. We are able to divide the 4-cell stage, if you do it very carefully with very fine needles and so on, to just really tease them apart gently. You can wind up with 4 separate cells called blastomeres, is what they're called, and each one of them can form a tiny. It's only about a quarter of the normal size, but each one of them can form a full functional larvae that can swim and feed and survive and sit down on a rock at some point and metamorphose into the adult urchin. Okay. And so that's what this schematic tells us. And when we look at the careful separation of these, and we can see that each of these cells has the capability of forming the entire organism. However, the next cell division, as I said, the third one, it creates unequal cells. So it's no longer a symmetric cell division. And what we have is an upper half. So if you take the 4 cells from the top, that's called the animal half. And if you culture it away from the vegetal half, you will get an animalized embryo. So it's like a blastula. It has just the ectoderm on the outside, and it has the cilia. And so it can swim around a little bit, but eventually it's going to die. It hasn't got any gut. It hasn't got any way of taking in nutrients or anything. So that's not a good condition. And then the vegetal half, the cells, can grow when they're cultured, and they will form gut and skeletal elements, exactly what we would expect based on the fate map that we see here. And again, this can't survive, it can't move, it doesn't have any ectoderm, so it's going to be missing nervous system, for example, and other things. And so this is vegetalized. However, what's really interesting, of course, they do all these different kinds of experiments, and this is a transplantation type of experiment. So if you were to take just the animal half of this embryo and culture it, you would get one of those animalized defective embryos. But if you take the micromeres from an earlier stage, this looks like the 32-cell stage, and this looks like the 16-cell stage. If you take the micromeres from the earlier embryo and you transplant them onto the animal half, they are the source of signals that allow for the whole thing to be repatterned, and we can get a completely normal Pluteus larva out of it. So that tells us that these micromeres have the ability to signal and send information to the cells above them and to tell them what kind of cell they're supposed to become. You can also do something similar to one of the experiments described in the early lectures, maybe lecture 3, if I remember correctly, where you have a normal embryo, and then if you take some micromeres from a different embryo, an earlier stage embryo, and then transplant it sort of to the side, so not in the normal place where micromeres are supposed to be, you can get the induction of, when we get to the gastrulation stage, we get the normal gut starting to form, but we get invagination and formation of a secondary gut. So this is evidence of organizer properties, right? So it's obviously telling these cells a certain kind of information, and then it causes, the formation of another gut here. And that should be familiar to you. It should sound similar to other experiments that we've talked about. So we know that those cells are sources of signaling, but we can't use sea urchin as a genetic model organism. We just don't have the ability to do it, and they're very hard to work with genetically. So the people working on this did something different. They looked at some other organisms, and they were looking at some work reported in amphibian embryos and we're just going to look at this one part because I'm going to talk about this again when I talk about frogs. Just the idea that, like, if you treat the embryos with lithium chloride, the lithium causes a hyperdorsal embryo. So an embryo that is incorrectly specified, it has mostly dorsal tissue and it's missing the ventral structures. And you might want to review wingless signaling, because it turns out that lithium is known to prevent GS3 kinase, which is part of the degradation complex, the destruction complex in the wingless pathway. And so if lithium blocks GSK3 kinase, that means we're not getting the destruction of beta-catenin, we're actually getting lots of beta-catenin able to be a transcription factor. And that means that this is acting like wingless signaling is on all the time. And so this is what we call the candidate gene approach. You find something that works a certain way in another organism, you do some experiments with your organism to see if that chemical treatment works the same way, and now you have this idea that, it's probably something to do with beta-catenin, let's take a look, right? And in the class, I don't think I have it in the net. No, I don't have it. But in the class, I'm going to show the results of some lithium treatment experiments and some other manipulations of beta-catenin, so that you can see how too much beta-catenin in the nucleus changes the specification in one direction and not enough changes it in the opposite direction. And so that's, be prepared to think about that when you get to the class. Okay, so this is a funny diagram, but I put this picture up here on the side so you can just kind of see structurally what we're talking about. This is the micromeres. This is endomesoderm, so this is tissue, some of which is going to form endoderm and some of which is going to form mesoderm. And these are the VEG2 cells, which are these here. And notice that, before cell division they have 2 fates, but after cell division these ones will form mesoderm and these will form endoderm. And then the VEG1 cells that are going to, after division, this part of them is going to form endoderm. So just to make it sort of physically, this is where we are, and then over time. So this is earlier in time. At the beginning, it's maternal factors that set everything up. There is Wnt signaling right from the the most vegetal region. There is a signal called early signal, which hasn't been identified. So it's just ES, and that is sent to the cells above. The micromeres themselves basically have maternal factors, and those maternal factors specify micromere structures, and they basically give rise to the skeletogenic genes. So these are the ones that are responsible for those spicule structures, okay, those skeletal structures. And then in the VEG2 cells, there is nuclear beta-catenin as a result of the signaling here. And TCF is the transcription factor, but that's not the most important thing. So what this does is, though, it turns on a whole bank of genes that are called endomesoderm specification genes. So these are genes that are needed to specify a dual fate of either mesoderm or endoderm. So they probably are turning off, for example, ectodermal fates as one of their jobs, and then beginning to activate certain genes that are needed in both endoderm and mesoderm. And then there is a second signal, and it is in this part of these cells, VEG2 cells. So it's Wnt8. It is being expressed, and it's signaling to itself. So these cells are signaling to each other, essentially, next door to each other, and they're signaling the cells above them, the VEG1 cells. So the signals, the Wnt8 signals are going in this direction, back and forth between these guys up top, and then also across to these cells. And so the cell, around here, there is actually a division, and we're now looking at two separate cells. These ones, because of the Wnt signaling, they have endoderm-specific genes activated, but those also are responsible for then turning on anything that is specific to just mesoderm, right? So you turn on endomesoderm first, and then when you turn on endoderm-specific genes, you also shut off the mesoderm-specific ones, and so you've got genes to form the gut. These cells, they have the nuclear beta-catenin, and so they turned on endomesoderm as well, but they're missing the Wnt8 signal, but they are getting from the micromeres also the delta signal. So there's no Wnt8, there's delta, and there's beta-catenin, and together beta-catenin and delta activate the mesoderm-specific genes, and they shut off those endoderm-specific ones. And so we have some additional mesoderm-specific fates. And these would be supporting cells around the skeletal elements and, whatever little organs there might be inside the sea urchin. And then the VEG1 cells, they don't have there's an almost undetectable amount of beta-catenin in the nuclei of these guys. So there might be a little bit of that signaling in here, but it will be very, very little. It's essentially none. And so just in response to the Wnt8 signal, they turn on also endoderm-specific genes. And they also are going to contribute to the formation of the gut. So hopefully that's clear. I'll go through it again in the class. Yeah, and we'll probably have a clicker question here or there. Maybe not on this, but on some of this related material. Okay, and so I just want to spend a couple minutes talking about the oral aboral axis. It's not exactly known what the initial, what we call symmetry breaking event is. So symmetry breaking event is a division where there's something in one of the cells and something different in the other cell, so that one of them is going to be oral and the other one is going to be aboral. We don't have sort of something that clear, but we do know that by around the 60 cell stage, we're seeing the expression of nodal in the oral ectoderm. So that's around the blastula stage. Nodal is a TGF beta, transforming growth factor beta family member, and its expression, it turns on genes like lefty and cordon, thought I changed this slide. This is an old slide, and I updated it. And BMP24, which is another TGF beta. Yeah, and there is a figure coming up that explains a little bit about that. Now, it turns out that if you make manipulations of nodal, you can figure out that it has quite an important function in organizing the whole entire oral and aboral ectoderm, and I'll show an image in a minute for that. If nodal is blocked, neither oral or aboral ectoderm are specified, so you have something that's indeterminate. It doesn't have, you know, the cilia and some of the other structures that are specifically associated with it differ somewhat, and so you can tell the difference between oral and aboral. And when you look at the ectoderm of a blastula where you've prevented nodal from signaling at all, it doesn't look like either one. It's just this sort of amorphous, unspecified tissue. Oops. And then if you express nodal in all the ectoderm, if you express it everywhere instead of just in the aboral side, then all of it gets converted into oral. So there's no aboral specified at all. And this is kind of how it works. So just bear with me. So early on, there is Wnt signaling from the vegetal side. So that's beta-catenin inside the nucleus, and that signaling moves upward and away from the vegetal side. So these cells signal to these cells, which signal to these, and so on. And you wind up with sort of endodermal fates, ciliated ectoderm, and the neural ectoderm is right at the top. So there's this kind of gradient of signaling that sends different information about the animal versus the vegetal signals. And then when we look at the blastula, which is really just one division later, that is when we start to see nodal being expressed. And nodal is expressed on the oral side. It activates the expression of other genes, and leptin and cordon are also signaling molecules, and they are found in the area that's going to actually form the ciliated ectoderm, sort of in this central region. And then BMP24, that's another TGF beta, so it's another member of the same family that nodal is in, but it's actually signaling on this side to form the aboral ectoderm. And so this is a little more, I'm okay if you learn this, because we're going to see lefty and cordon and BMP constantly in the other kinds of, when we look at frogs and mice and that sort of thing. But if you just remember this, that will be good enough for me, so that there is a Wnt signal from the vegetal side that creates a kind of vegetal animal gradient, and it turns out that these signals are very similar to posterior versus anterior in other organisms, and that there's a nodal signal on the oral side that you know, via various mechanisms leads to BMP24 on the aboral side, and that these correspond roughly to ventral versus dorsal in other organisms. So we've got different names for them because there's really no ventral or dorsal in this organism. It's round, so it doesn't have a front and a back, but it has an axis that still has the same features, and so that's part of that. Okay, so I've got a summary slide here, and I probably haven't put everything on there, but I'll maybe update it before I post it for you. And the next lecture is going to be on plants, and we're going to spend 4, I think, lectures talking about all the really cool things that plants do.